Let's start the last couple chapters of your organic chemistry experience. We're going to talk about carbohydrates. This is a section that covers biomolecules. We'll cover proteins, nucleic acids, and a little bit about lipids. So first of all, we need to introduce you to carbohydrates, classification of monosaccharides, and then we'll continue on into what are aldoses, ketoses, and monosaccharides, and along down that route. So, carbohydrates are bioorganic compounds. They're found in biological systems. What's new about them is these are larger and often more complex structures. So they have multiple functional groups, and that's important for recognition biologically. So your body needs to be able to recognize different molecules and what they are and what they're used for. So we will have some reactions, but they're actually reviews of reactions that we've seen in the past. So this is actually a good chance to review several of the reactions we've talked about previously. Now, carbohydrates are the most abundant class of compounds in the biological world. If you took all the biomass, meaning all the things made biologically, and you dried them out, carbohydrates would make up greater than 50% of the dry weight. So they're important in cell surface recognition. So in fact, fertilization occurs when the sperm recognize a carbohydrate on the egg cell. In fact, viruses and other bacteria will also recognize carbohydrates on the cell surface. So they're metabolic fuel sources, meaning that plants and animals actually use them for energy. And so carbohydrates arise when plants take carbon dioxide and water, take sunlight and oxygen from the air, and combine it and make glucose. Glucose is then extended into longer polymers. They're known as cellulose and starch. And these are used, cellulose specifically, as a structural component of plants and starch as food. So the other biomolecules that we'll talk about are amino acids that include peptides and proteins. We'll talk about nucleosides, nucleotides, and nucleic acids, and lipids. But this chapter focuses specific on carbohydrates. So, introduction to carbohydrates. So the most abundant carbohydrate in nature is glucose. And as I mentioned earlier, plants make glucose and animals take the glucose, and then we take oxygen from the air and we produce CO2 and water. And I might have misspoke earlier. So, sorry, plants take CO2 and water and make oxygen and glucose. And animals, humans in this case, do the reaction in reverse. We take glucose and oxygen and give back the plants carbon dioxide and water. And so this is a process referred to as oxidation. We're using oxygen to do this, and it provides energy when needed. Now, we don't just store our glucose as just individual molecules. We tend to store it as longer chains. In animals, it's known as glycogen. In plants, it's known as starch. And in cellulose, but that's not really an energy storage, that's more of a structural component. And so these are also found in crustaceans as structural components, in chitin. These are modified glucose that have actually an amine in there. So this connection between plants and animals, plants use photosynthesis to make the food. We eat the food, make the carbon dioxide and water that the plants need to make glucose and oxygen. So a very symbiotic relationship between plants and animals. Okay, so carbohydrates, saccharides, sugar, they're interchangeable terms. They're classified by the size, whether they be monomeric or polymeric, meaning multiple. So simple carbohydrates tend to be monosaccharides or simple sugars, and this just includes a single sugar molecule. And then you have complex carbohydrates that contain either two of these individual saccharides or carbohydrates linked together. Yeah, if there's just two, we refer to them as disaccharides. If there's 3 to approximately 10, they're oligosaccharides. And if there's 10 or more, or more than 10, then we refer to them as polysaccharides. And so how you can classify them is, well, if you just had a monosaccharide and you introduced water, nothing would change. There'd be no hydrolysis. A disaccharide, if you introduced water and a little bit of acid, you'd end up getting 2 monosaccharide units. And if you had many of them linked together, in the case of a polysaccharide, and you added multiple water and again acid catalyst, you'd get many monosaccharide units. So these disaccharides and oligosaccharides and polysaccharides can be broken down into their individual units, their monomers, or their single units. So this is M for either monomer or monosaccharide unit. And again, a polysaccharide has them linked together in very long chains, and we'll talk about that actual linkage soon. And so to break apart that linkage, it's simply a hydrolysis reaction, and you end up with your individual monosaccharides. Let's talk more about the structure of carbohydrates. They have roughly as many oxygens as they do carbons. So we would say that they're fairly highly oxidized molecules. You can oxidize them further. In fact, that's how we get the energy out. We oxidize them ultimately to carbon dioxide and water, but they're already started. They've got some handles on there that we can use to further oxidize. Now, if this wasn't oxidized at all and was just a alkane, us as humans would have a very hard time metabolizing that. There's no functional group handles for us to actually grab onto and do further chemistry. So the molecules that we use for energy actually have handles and they're partially oxidized. Now, the reason that they're called carbohydrates is if you look at them, roughly their empirical formula has a carbon and an H2O. So for every carbon, there's roughly 1 H2O. So it looks like it is a hydrate of carbon. So a C with a H2O. So when they first started looking at this, they noticed that relationship that for every carbon, there's roughly 1 water. And so that's where the name came from, as a carbon hydrate. So currently, though, they're natural molecules that contain many hydroxyl, so they're polyhydroxyl molecules. So many alcohols on these. And we can represent them in many different ways. We learned previously about the Fisher projections, and this is where the Fisher projections really shine in helping us represent sugars. Now, if you look up at this top molecule, which with all the wedge and dashes, it gets pretty cumbersome, and it's hard to actually see the stereochemistry. We could draw them in this wedge and dash form over here, but we can simplify that. So what we use is a Fisher projection, and the Fisher projection typically takes the highest oxidized group and puts it at the top. And then the backbone is actually just vertical, going up and down. And then these groups here are horizontal. Now there's a convention. Those groups that are horizontal are actually pointing out at you, which means that the backbone is actually pointing away from you. If you actually made a model of this, you'd actually see that the backbone actually curls the molecule away from you. So we can't just really make a molecule straight because we know the bond angles around carbon is 109. But this does allow us to represent the carbohydrate in a very simplified form with, again, the understanding that this vertical is rolling away from us, and anything that is horizontal is pointing out at us there. And so how we come up with the Fisher projection, essentially we just look at it like we're squishing it flat. So again, it looks like the bond angles are 90 degrees, but they're not in the real molecule. This just allows us to see how the molecules are arranged. And there's a couple of things when we put molecules in Fisher projections that we can and can't do. One thing that we can't do is rotate them 90 degrees. If we just took a Fisher projection and rotated 90 degrees, actually the stereochemistry would change around each of these chiral centers. Now you can rotate them 180 degrees, meaning just flip them upside down, but you can't rotate them 90 degrees. Those are not equivalent structures. Okay, now when we have these carbohydrates, we actually have ways to classify them. So we've got lots of vocabulary here. So we can classify our carbohydrates or our sugars by the type of functional group that they have, or the highest oxidized carbon. In this case, the highest oxidized carbon is a C double bond O, so a carbonyl with an H off there. So that is an aldehyde functional group, but we're going to name this as an aldose. So essentially an aldehyde sugar. The ose comes from the term for the sugar, and the aldehyde because it has an aldehyde in it. We can also have what are referred to as ketoses. So this has a ketone, again, polyhydroxylated, and so it has the ending ose. So that's the general classification, is aldose and ketose. This one has the specific name glucose, and this one here has the specific name of fructose. For right now, you should be able to develop the skill of generally classifying a sugar as if it's an aldose or a ketose. Now we can further classify the sugar based on the number of carbons. So if it contains 3 carbons, it's referred to as a triose, 4 carbons, tetrose. And then once we get up to 5 carbons, we use the prefixes that we used before, pentose and hexose and heptose and octose and nonose. And so we can actually go and classify the sugars a little bit more. We can actually call it an aldo. In this case, we count the number of sugars or carbons, 1, 2, 3, 4, 5, 6. So there's 6 carbons in this sugar. And so we would classify it as an aldohexose. That's its generic name. So glucose is an aldohexose. And fructose would be a keto. And let's count up the number of carbons, 1, 2, 3, 4, 5, 6, a ketohexose. Now that's not the only classification that we can do. There is some stereochemistry here that's of importance. This very last stereocenter right here is very important, and it classifies the sugar as either D or L sugars. So in this case here, that OH is off to the right, and that is what's referred to as a D sugar. And nature prefers the D sugars, and for some reason, it's this very last stereocenter. So if we draw this with the highest oxidized end up at the top, and we go down, this is the last stereocenter there, and this last stereocenter right there. Again, if that OH is off to the right, it's referred to as a D. If it's off to the left, it's referred to as an L sugar. And this comes from when they first started classifying the simplest sugars. These are aldotrioses, known as glyceraldehyde. Only one stereocenter, Again, if we come from the top and we go to the very last stereocenter, of which there's only one in this molecule, and we look, the OH is off to the right, and that is referred to as D-glyceraldehyde, and this one is off to the left, which is referred to as L-glyceraldehyde. And so we have to use the Fischer projections to actually classify these sugars as D or L. Again, placing the highest oxidized group at the top and drawing them down, and then finding the last stereocenter, chiral center here, and figuring out if that OH is pointed to the right or the left. So again, we can classify these sugars generally. And so you can see this example here on the left is an aldopentose, and specifically a D-aldopentose. You can look at the molecule on the right, and it is a ketohexose, and specifically a D-ketohexose. And so again, what we find out is that nature prefers the D configuration down here. So that is pointed off to the right, and so that is referred to as the D sugar. And these are all D and natural sugars: glucose, fructose, ribose, and seduhepulose, which is referred to as a ketoheptose. And again, these are all referred to as D sugars. Okay, so D and L sugars. So glyceraldehyde exists as two enantiomers, first identified by their opposite rotation of plane polarized light. And so when they first started studying sugars, this molecule right here rotated light dextrorotatory, or to the right, and this was levorotatory, or to the left. And that is where this D and L comes from originally, is how it rotated the plane of polarized light. As they started to understand sugars more, they realized that there was this commonality, that most natural sugars had this configuration here at the very last carbon. It was pointed to the right when we drew it in the Fischer projection. And so now the D simply corresponds to the configuration of that last stereocenter. So this is D-galactose, and this is L-galactose. So these are specific sugars. Notice in L-galactose, everything is a mirror image. Each one of these OHs is opposite, as in a mirror. So we to go from D-galactose to L-galactose, we just don't switch the last stereocenter or chiral center, we switch every stereocenter there. So they have to be complete mirror images of each other. So as they went and studied sugars more, they realized that the rotation of the plane polarized light doesn't always correspond to just that last stereocenter. It does for D-glyceraldehyde and L-glyceraldehyde. Okay. So that's where the D and L originally came from, but no longer does D and L correspond to the way that it rotates plane polarized light. You may remember this when we talked about chirality. You put the sample in the sample tube and you shine plane polarized light, and then you look at it from the other end. And this plane polarized light only allows light to come through in a certain orientation. And so what you have to do is rotate the analyzer until you can actually see the light. If this analyzer, the slits were straight up, it would just appear dark. So we rotate it until you see the light, and that gives you the angle that the molecule is actually rotating this plane polarized light. And so again, for glyceraldehyde or D-glyceraldehyde, it rotated it to the right. L-glyceraldehyde, it rotated it to the left. And it looks like they're finally here to vacuum. So I'm going to have to pause for a minute till they're done. Okay, high production value. I've got a clean office now. So we're talking about how we classify D and L sugars. Again, originally it was based on the And now my computer froze.